Welcome back to looking at the effects of different methods that can be used to control microbial growth. And in the first part, we're going to focus on this area here with our physical agents, and we'll briefly take a look at the mechanical methods to remove microbes. And finish up with these different types of chemical agents. And again, when you're looking at the effects of these different kinds of methods, you have to think about the difference between disinfection and sterilization when you're using this kind of techniques on inanimate objects or fomites, and compare that with the ability to do antisepsis using different kinds of chemicals on living tissue that's out there. And if we look at this first side over here with our physical agents, heat is definitely one of the methods that can be used to remove microbes. But again, you have to be careful. You might be able to use heat on a person, but you're going to cause more damage to their tissues than you probably are going to cause to the microbe that's causing a disease. So there is a time and a place to use different methods of heat to control the growth of these different kinds of microbes that are out there. And when you look at something as simple as using your flame on your loop or burning things out there, really what you're doing is the technique of incineration is the ultimate means of controlling microbial growth, because you are oxidizing the organic molecules in those cells and turning them into CO2, and that is not going to be able to support life after that. If you put things into an oven and you dry them, you're going to drive away that fluid in the cytoplasm, and you're also going to cause damage to a lot of the cellular structure. So both of these can be used to get rid of the microbes that are out there. Certainly with the dry oven, it's going to take a longer time compared to incineration to get rid of the fluids that are available in the cells. If we go up to a different method of treatment, we can go from dry heat to moist heat. And the thing to consider here is if you look at that idea of moisture that's been heated up, you're taking your water and you're putting a bunch of heat energy into it and increasing the kinetic energy of the water. That's going to cause your water molecules to spin around faster and faster until eventually you reach a point where they collect enough of that kinetic energy that they can get away from the liquid and break all those hydrogen bonds holding them there, and now you transfer the water from liquid to vapor phase, and that steam that is coming off of there has a huge amount of kinetic energy. And when that comes in contact with cells or your tissue, and the water goes from the gas phase back to the liquid phase, it's going to release that kinetic energy. And so again, that's going to cause damage to the proteins in your cells. It'll denature them. It can cause damage to your membranes, which again are these fluid mosaic chemicals that have your phospholipids and your proteins and whatnot floating around in there, and so that'll cause damage to those membranes, and especially sensitive are your eukaryotic cells with their internal membrane structures. And if we go all the way up to an autoclave, we're going to put our water under high pressure, typically about 15 pounds of pressure per square inch. To give you a comparison, your tires are probably around 30 psi, and this is going to be a lot higher pressure. And under higher pressure, your steam is going to be able to boil at a higher temperature there. So as opposed to water boiling at 100 degrees Celsius, now you're up to 122 degrees Celsius. So that's going to penetrate really far into things and cause a lot of damage to the cells. And if you do it for long enough, you're going to sterilize most of the kinds of microbes that are out there. But again, remember those exceptions. When you're looking at endospores or you're looking at things like prions, they are really resistant to this method of treatment. We can also use moist heat in less severe kinds of conditions over here to disinfect. And this is a good example of using moist heat, is the idea of pasteurization. And when you look at this idea of pasteurization, you're really looking at the ability of heating these things up enough to a point where you reduce the number of microbes that are out there. And you can do what's known as a batch method, where you treat massive amounts of liquid And so you could see something where you have gallons or thousands of gallons of milk. You heat it up and hold it at 63 degrees Celsius for at least 30 minutes, and that's going to reduce the number of spoilage organisms and pathogens that are in the milk to the point where they should not pose a danger of causing a disease or the milk going bad quickly. But again, it's not sterile. This is pasteurized, so you've reduced the microbial load, and there's still potentially going to be some left over there. But depending on the types of microbes, a lot of them in their vegetative phase are going to be sensitive to even just a simple 63 degrees Celsius heat treatment for 30 minutes. If you go up to higher and higher, more severe kinds of treatments, we can look at these two different versions right here, and here you can see this pasteurization technique where you do have to keep your stuff refrigerated there. And if we go up to ultra high temperatures, now we're going to be heating our milk up to at least 138 degrees Celsius for 2 or more seconds, and then you seal these guys up in containers, and these should remain free of the microbes that are out there for at least 90 days. And notice you don't have to use refrigeration for this. You can also have a form of ultra high temperature instead of pasteurization. It's known as UHT sterilization. And here you actually take it, and not only do you heat it up, but you quick chill your milk down. And this is going to prevent membranes from reforming and prevent proteins from folding up effectively. And so you can see the difference if you look at creamers in restaurants that are out there. If it's pasteurized, they're supposed to keep them on ice to try and reduce the chances of growth. If it's sterilized, you don't have to worry about anything growing up inside of these little guys. Likewise, if we can go to the hot side, we can also look at the cold side for temperatures. And oftentimes when we look at the effects of lower temperatures out there, this actually is going to be used as a preservative technique there. Freezing, especially with eukaryotic cells, they tend to be more sensitive because the ice crystals that form are going to punch holes in the membranes, and so eukaryotic cells are often more sensitive. And this would be used with something like sushi, where you take your fish and you put it into the freezer, and then you thaw it out, and then you put it back in the freezer and you thaw it out. And so that can be used to actually go through multiple freeze-thaw cycles and cause damage to pathogens that might be in the fish there, and reduce the microbial load that's there. But when we look at bacteria, freezing tends to be bacteriostatic and not cytal. And so bacteria can often survive as frozen cultures, and here we can see they can be stored at minus 80 degrees Celsius in a normal lab freezer. Also for long-term storage, a lot of people like to use liquid nitrogen, which is going to freeze at a much, much lower temperature. You can see almost down to minus 200 degrees Celsius there. One of the interesting things also that people have found is a lot of bacterial cultures can be stored in what's known as a lyophilized state. And what you do with this is you actually freeze the culture and you dry it under a vacuum. And under a vacuum, water will actually boil at room temperature. And so you can literally freeze dry your culture and get rid of all the water that was in the cytoplasm and in the culture media, and you're left with basically salt and dried up husks of all this organic material. And this really mimics the natural state that you can find in a lot of the environments for bacteria. So imagine in the summer around Merced, it's going to be very dry, and so the cells have adapted to growing or to going through this seasonal change by basically being able to go into stasis and wait until the rains come back later on. And so what people have found is they can take lyophilized cultures and they seal them up in these little glass vials like this, and you can store your bacteria culture for literally decades, even at room temperature here. And then when you want to recover your culture, you just add some water to it, and your cells will then wake up from their stasis, and you have a live, viable culture. And so again, when we think about this, think about the danger of especially spores that are out there. When they run into some water and the right environment, those cells are going to be able to wake up and go back and germinate into the vegetative state. So it's not necessarily used as a method to control growth so much as just stopping growth by freezing things down here. One of the other easy methods that we can run into out there that relies on a physical characteristic is the ability to add things like different kinds of salts or solutes, like sugars, to a culture, and the more of that stuff you have on the outside of the cells, the more osmotic pressure you're going to cause. And that hypertonic environment, that's where the water is going to go. So literally when you're taking your beef jerky or the fish or the jam that we see here, by the addition of the solutes to the outside, you're going to desiccate or dry out those cells there. And the lack of that cytoplasm is going to result in the death of the cells there. Again, spores are going to be able to resist this because they're not metabolically active and they aren't dependent on the presence of water to maintain the structures for the cells. The last method we can really look at as a physical method for controlling growth is using radiation to kill your cells off, or to at least cause enough damage to your cells that the cumulative effects of that result in the death of the cells. And when we look at something like ionizing radiation, this is really a stronger form of radiation that can penetrate much further. So think X-rays or gamma rays. And a lot of foods are going to be sterilized by using ionizing radiation, where you actually are exposing things like spices to a radioactive isotope. Oftentimes they will use cesium blocks, and then you keep your food in close proximity to this, and the gamma rays penetrate through the food, and they're going to cause damage to the DNA and break it up or fragment it into little bits and pieces, and that is going to result in the death of the cells because they cannot put all those little pieces back together to reassemble their chromosomes. When you look at non-ionizing radiation, we sort of saw the results of this in the lab, because if you look at the growth of Serratia marcescens in the presence of ultraviolet light, the majority of cells die off, and the longer exposure there is, the more likely you are going to have that damage to the DNA where you form that thymine dimer. And then when the DNA is used, you make the wrong RNA because you skip that region where the DNA is bent. Or when the DNA is used for DNA replication, it's going to result in a deletion mutation as your DNA polymerase skips where that kink is in your DNA. The problem with this non-ionizing radiation is UV light cannot penetrate through solids, so you might be able to sterilize the surface of an object, but you wouldn't be able to sterilize the interior because the UV light cannot get in there. All right, the last one we can see here for our physical methods is this idea of using filtration. And with filtration, what you're effectively doing is using filters that are going to have small holes or pores in them that will allow certain size particles to get through. And when we look at a HEPA filter here, it's actually a stack of a variety of different kinds of filters. And these are known as high efficiency particulate air filters. And so you can see, if you look at this figure here, that some of the larger pieces or chunks of things floating around in your air are going to get blocked by this filter, but smaller ones will go through, and those are going to get intercepted as they work their way through the filters, and that's going to prevent these particles from getting through. So effectively, your filtration is removing those particles from the air, or over here we can remove them from liquid. So imagine you're using some sort of culture medium that has sugars in it or proteins in it. If you put them in the autoclave, you can sterilize it, but you're going to denature those, or you're going to break down the sugars, and they're no longer going to be available for your cells to use. So by running them through a membrane filter with really small pores, you're going to remove those particles from there and effectively decontaminate that solution and remove them. But this does bring up the question, if you sterilize media so that you have a filter that does not allow bacteria cells into it, you could potentially still have viruses there, because those are smaller than your cells and they can possibly travel through the filter that you're using there. And so that begs the question, is this really considered sterile media? Well, it's definitely sterile in terms of microbial life, but it's not sterile in terms of acellular particles that might get through your filtration mechanism. So remember, if you look in your textbook, you can see this table that goes in to summarize all these different methods that I talked about. And this is really a good thing to take a look at for the test, because it shows you a lot of the different kinds of conditions where you want to use these things, how they actually work, and then provide some examples for where you use some of these different techniques.